So like I was saying, fungi can be used to produce food, produce antibiotics. For us, we use fungi in the form of yeast to make wine products, to make alcohol, to make bread, to make pizza. Really can't really get away from using yeast, which is a type of fungus. The molds, for the most part, we don't really use them for things that are edible. Or things that we like to eat, but we do use them for antibiotic treatments, and we use what's known as fleshy fungi, which are things like mushrooms, so that we can enjoy and eat in our salads and on our pizza, and as kind of a vegetarian option for some folks. Now, out in the wild, fungi play a very critical role in serving as decomposers. They are known as to being what's referred to as saprophytes, which means they help to digest organic material by decaying it using enzymes that are expressed by fungus. Fungi have some similar attributes to plants, but they are not plants. They are classified in their own kingdom. They aren't plants because they are considered to be heterotrophs, which means they have to digest organic compounds, and they don't have any chlorophylls, so they don't generate energy from the use of sunlight, and they don't undergo the process of photosynthesis. They don't have any roots to them, and they don't have any leaves, they don't have any stems. So fungi are not considered plants, they're in their own kingdom. Some other characteristics that fungi share is the fact that in the cell wall of a fungal cell, there is a specialized type of polysaccharide that is known as chitin. And chitin helps to provide durability to the durability to the cell wall of the fungus. It also helps to retain water inside of the cell. There are some fungi that are going to be unicellular, kind of like the yeast. The yeast are pretty much the #1 fungi out there that can exist as one cell. But others grow and are mostly what's referred to as filamentous fungi. They have special filaments that grow all throughout their body. That's known as hyphae. And hyphae is known in the collective sense as a mass of mycelium. And mycelium, which is made up of hyphae, provides fungus with structure and support and protection, and it makes up the bulk of the organism. So think of hyphae as kind of like fibers that hold together a mushroom. And then the giant mass of these fibers is known as mycelium, which will be fitted all throughout the body of the fungus. Fungi can be classified as to whether or not their hyphae is going to be septate or aseptate. What this means is that the hyphae themselves, which are essentially just cells, they're going to be divided by each other through the existence of what's called a septum. And a septum is just a wall that you would see between two fungal cells or between two hyphae. And what's interesting about this wall is that in most fungi, there's a little pore that exists through the wall, and that little pore can allow the flow of the nucleus in the fungal cell to pass through it. So sometimes you might see two nuclei situated in one fungal cell because the nucleus has passed through the little pore in the septum. Now, not all fungi have septate hyphae. Some fungi have aseptate hyphae, in which there is no septum, and then it just looks like you have multicellular, or you have multinuclei incorporated into one single fungal cell. So it's kind of interesting. But these key characteristics help us to classify fungus based on whether or not they have septate hyphae or aseptate hyphae. So we use that for identification purposes. fungal cells, in terms of reproduction, undergo reproduction, and they can do two ways. They can either do asexual or they can do sexual. And then in some fungal species, you can have both asexual and sexual. So the two categories of the generation of spores, which are what fungi use in order to pass on their genes, are going to be either sexual spores or asexual spores. If you have asexual spores, you have a special scientific term associated with them that's called conidia. So anytime you hear conidia, associate that with asexual spores. And like I said, some fungi will produce both asexually as well as sexually. Spores are really great structures because they help to protect the genetic material that's encoded in them, very much like the endospores that we talked about with the bacteria. But remember, in bacterial species, the endospores are considered to be not related to reproductive success. With fungus, though, this is the first time in a different kingdom where we see spores being generated that allow genetic material to be passed on. And when those spores are generated, what they do is they land in an environment, and in some instances there's male spores and then there's female spores. They meet up and then they produce full-blown fungus. Or in some instances you just have the release of the spore, and then the spore will just start to germinate into a new fungal organism. So here's kind of a look at those diagrams of the kind of key terms I've been talking about. So here's an example of hypha. Remember I told you they were like a hypha, or a hyphae is a thread-like material that makes up the bulk of a fungus. And then you have hypha being separated by these septums. Now if there is no septum present, we call it aseptic hyphae. And then you can see all of these nuclei that just look like they're in this fluid matrix all by themselves. So all the cell barriers, all the cell septums have disappeared, and that looks just like a free streaming location for nuclei to be situated. Here is a kind of an example diagram of yeast growing on probably an agar plate. So here's the petri dish, and here's the culture medium. You can see the yeast colony is growing on top. The yeast is going to look very similar to bacteria, but when you look under the microscope, the structures will be much different. Bacteria will be either coccus or bacillus, whereas the yeast are going to be these giant balloon-like structures that look like small little buds if you looked at them under the microscope. They don't reflect a morphology that's similar to bacteria. That's how we know that they're fungus, because they're bigger. For this organism here, this is some mold that's growing on the plate. In the image, it looks just kind of like a plant. That's because of the colors that are used. There is some mold that you would see, like penicillin, that grows green. And then you can see that you have this hyphae that is serving as an anchoring point, as it's anchoring the organism to the culture medium in which it's growing in. And then you also have these aerial hyphae, which are going to, at the end of them, you can see these black little dots, which is where the spores are going to be formed. And so down here you have hyphae used for anchoring and absorption of nutrients from the culture media, but up here you have the aerial hyphae, where we have essentially the reproduction structures for the fungus. So up here you can have the generation of spores, and then the spores will get released into this petri dish. And once they're released, they'll land on the agar, and then they'll create vegetative hyphae as well as aerial hyphae and allow the organism to reproduce. So that's pretty interesting, and it takes about 7 days for mold to generate, between maybe 3 and 7 days. So in my laboratories, we generate them within the span of 2 days. So it kind of depends on the inoculum that you use. Fungi classification is based on how fungi are able to reproduce, either through sexual reproduction or through asexual reproduction. And then if a spore is being produced and reproduction, then typically that's going to be an asexual a reproduction kind of pattern that you would see. There are 5 distinct phyla of fungi. There's the Zygomycota family, the Chytridiomycotia family, the Ascomycotia family, the Basidiomycotia family, and the Deuteromycotia family. And the Deuteromycotia family, these are also known as Deuteromycetes. They include some very dangerous types of fungus, which can be respiratory irritants, such as Aspergillus, which is a genre of fungal organisms that cause respiratory complications. But they can also include organisms like penicillin, which we use pretty extensively for antibiotic treatments. The organisms that make up this particular phyla, the Deuteromycetes, they don't have a mode of sexual reproduction or a mode of the mode of sexual reproduction is not very well known. So they're still doing research into kind of clearly understanding how it is these organisms reproduce. And for the most part right now, it's all leading back to asexual reproduction, not sexual reproduction, where you have the fusion of male gametes with female gametes to generate spores. So instead, we're looking at asexual reproduction, in which penicillin will release spores that contain its own genetic material to generate identical organisms. And here's some images of some of the fungus that you can see in the kingdom. Aspergillus fumigatus, you can see it has these interesting little conidias, which are at the end, these little hand-like structures. This is called the thallus, which is the stalk of the fungus, or in a sense you also refer to it as the hyphae or mycelium. Here's penicillin. These round little structures are referred to as the spores. Aspergillus flavus has a different shape from Aspergillus fumigatus. And then Curvularia, Histoplasma capsulatum, and then Scopularia opus, which is located here. So we do have, kind of a good variety of different fungus, which is interesting because when you do a fungal stain and put it on the microscope, or you're taking a clinical specimen and viewing it under the microscope, you may be thinking that a patient's infected with a bacteria, but then you see these elements and you're like, oh no, it's not a bacterial organism. These are a lot bigger, more complex, and these are all going to be fungal organisms that have infected a patient. So that's why we're discussing this, so you know what to look for if you had to look for these organisms under the microscope. Yeast are unicellular. They're kind of the most boring of the fungi, in my opinion. We use them extensively because we like to eat them, or we like to prepare food using yeast. But with yeast, they're interesting because they don't have any mycelium to them. They just look like a very small round balloon or oval balloon. The yeast cells themselves have some distinct terms that are associated with them. You can refer to a yeast cell as a blastospore or a blastoconidia. And then these are pretty much the only fungus where you can really completely see the organism underneath a microscope. Yeast typically undergo the process of asexual reproduction, and they use the process known as budding, where you have one yeast cell and then it will just bud into a new yeast cell, so you have the formation of 2 yeast cells when that takes place. And when you have a lot of buds that start budding, a special term is applied to understand these, and these are referred to as pseudohypha. Because they're just long in length, but they don't really have a hypha associated with them. Yeast also contain a specialized structure that's very thick, called a chlamydospore, and this helps to serve for protection as well as retaining of water inside of the cell, so that way the cell is able to survive its current conditions. Now here's an image of some budding yeast cells that you can see. It's a gram stain clinical specimen, so I guess they were looking for maybe some bacterial cells, but what they ended up finding were these budding yeast samples. And the